Well, good evening, everybody, and welcome. My name's Dave Petley. I'm Pro-Vice-Chancellor for Research and Enterprise here at UEA, and welcome to this inaugural lecture, and it's wonderful to see so many of you here. So it's one of the great pleasures, I think, of my role as PVC here to introduce inaugural lectures, to hear the great research that our colleagues have done over the course of their career and, of course, have continued to work upon. I'd like to particularly extend a warm welcome to those who have joined us today from outside of the university. It's great to see you here. You're in our new Enterprise Centre. This is UEA's newest building. It's our greenest building, and it's a building of which we are immensely proud. This centre has been designed and built to help us to harness the creative thought and innovation which comes out of our research, to take the deep understanding we cultivate and to apply it to the world outside of academia. And the way it operates is it encourages both students and our active researchers and people who are engaged in enterprise activities from outside of the university to meet, to congregate, and to develop ideas. And as tonight's lecture will demonstrate, the work we undertake at UEA has the potential for genuine real-world impact. I'm delighted to be introducing our Vice-Chancellor, Professor David Richardson, to speak this evening on his own area of research, which is microbiology. David became UEA's Vice-Chancellor in September 2014. He joined the university in 1991, and since then he's held a series of leadership positions, including Dean of the Faculty of Science, Pro-Vice-Chancellor for Research, Enterprise and Engagement, and Deputy Vice-Chancellor. As a member of the Governing Council of the Biotechnology and Biological Sciences Research Council, he plays a key role in shaping UK research policy. David's own research career as a microbiologist began at the universities of Birmingham and Oxford. He has led a research group in UEA's School of Biological Sciences for 23 years, conducting world-leading research into the biochemistry of environmentally and medically important bacteria. In 1999, he was awarded the Fleming Medal and a Wolfson Royal Society Fellowship for his contribution to the understanding of bacterial bioenergetics. As many of you will be aware, UEA is one of several institutes, laboratories, and other facilities located here on the Norwich Research Park. And this also includes the Norfolk and Norwich University Hospital, the John Innes Centre, the Sainsbury Laboratory, and the Institute for Food Research. The park has become a real research powerhouse over the last few decades and is now home to some of the UK's most highly cited researchers. UEA's partnerships across the park give our academics the opportunity to collaborate with other leading experts across a range of disciplines, including health, food science, and environmental research. Not only does this lead to new discoveries and the publication of internationally recognized research, it also expands the opportunities available to our students to gain practical experience of working in world-class facilities. We are immensely proud of the work that our colleagues undertake here at UEA in finding solutions to global challenges, and that will be a major focus for us in the coming decade or so. For example, we work in the study of nutrition, on our aging population, on water security, on climate change, on international relations, and in many other areas. David's own research group seeks to address some of the greatest challenges we face today, including the management of increasing pollution, securing food for our expanding population, and the growing need to identify sources of sustainable energy. So please now join me, without further ado, in welcoming Professor David Richardson to give this evening's lecture on greenhouse gases and bioenergy. Thank you.
Well, thanks very much, Dave, for that very generous introduction, and thank you to you all for coming out this evening on what is quite a blowy day around Norfolk, although this lecture's been streamed live, so for those who aren't in Norfolk, it's lovely and sunny here this evening, because it always is in Norfolk, of course. I have been here for 25 years, and I've actually been a professor for 15 years. And a number of people have asked me, is this really your inaugural lecture? You must have given one. And the truth is, it really is my inaugural professorial lecture. When I was promoted to chair 15 years ago, I did manage to escape giving an inaugural, but of course, they always know where to find you, and you can never escape the UEA events team. So eventually they found me, and they thought it would be a good idea, and I did too. And it really is a great pleasure to get the chance to tell you all a little bit about the research area in which I work, and also about the research that we are continuing to do in my group and in the groups of my collaborating colleagues around UEA. So whilst I've been here 25 years, I very much want to make this lecture a prospective look at the challenges that we're going to be addressing in the future, rather than too much of a retrospective look over what I've done in the 25 years here so far. So, greenhouse gases and bioenergy, theologians and radicals having a laugh while Martians are eating metals. All a little bit cryptic perhaps, and hopefully it will all unravel as we go through the next 45 minutes or so. But my work over the 25 years has focused on a process called respiration. And the place to start then is to introduce you all to what that process actually is. And so, if you just focus on yourselves for a little bit and just think about your breathing, and you just start to realize that you're taking air into your lungs and you start relaxing a bit as you're doing it. And if you're anything like me, you immediately worry that you're going to stop breathing. And you won't, you'll be fine. So just bring it in. And what you're actually doing there is you're bringing oxygen into your body. And that oxygen is being respired. And in that respiration process, in quite simple terms, what we're doing is we're burning up organic carbon, the organic carbon that we ingest in our diet, and extracting electrons from that organic carbon and passing it to oxygen, an electron acceptor. In many ways, setting up an electric current, and we'll see how we can harness those electric currents later on in the talk. Now, in that process, energy is being released, and energy can be released as heat. We've just seen that fire burning away there. And that heat is what keeps us alive, what keeps our bodies at 37 degrees centigrade. But also some of that energy is captured as chemical energy. Chemical energy within the cells that enables the cells to grow, to survive, our bodies to repair themselves. And it's essential, of course, therefore, for life. And that process of respiration is taking place in our bodies all the time, of course. And it's how we, as humans, survive. And so respiration takes place across a large scale, from meters through to nanometers, meters through to 10 to the minus 8 meters. And actually, at UEA in the Norwich Research Park, we study respiration across all of those scales. So, of course, we take that air into the lungs, and then once in the lungs, the oxygen goes into the bloodstream, and eventually will enter cells. Once the oxygen's in those cells, it actually goes into little organelles, compartments within the cells that are called mitochondria. And in those mitochondria, the oxygen will ultimately actually combine with electrons in what we call electron transport or respiratory proteins. And now we're at the 10 to the minus 8 meter scale, the nanometer scale, and it's that combination of oxygen with electrons extracted from organic carbon taking place here in these proteins that finishes that respiration process and releases the energy. And across the Norwich Research Park in UEA, we do have people looking at all these scales. I think I saw the Dean of the medical school walk in a few minutes ago, and he's actually a mitochondrial biochemist, and many diseases are associated with inborn errors of metabolism based around respiratory systems in mitochondria. Now, for my part, I don't work on mitochondria. I work on bacteria. You've heard that I'm a microbiologist or a microbial biochemist. And these mitochondria, maybe 2 billion years ago on this planet, actually evolved from bacteria. And so those original respiratory processes taking place in our cells, in our bodies, in these things called mitochondria, actually evolved in bacteria that are similar size to these mitochondria, but evolved on this planet billions of years ago. And those bacteria, when they were evolving on this planet, didn't actually have oxygen to respire because there was no oxygen on our planet 2 or 3 billion years ago. The oxygen came about 2.5 billion years ago. So 3 billion years ago, when these microbes were evolving, it wasn't there. And so there were other respiratory substrates, respiratory electron acceptors, that had to support life, that had to drive life in the absence of oxygen. And one of those early ones, perhaps the earliest one, was actually metals or metal oxides. And here we have, oops, sorry. Just get this going across here. I've got a little video. And this is a metal being respired in the way that we respire oxygen. It's a mineral of manganese oxide. It should have a half-life of about 14,000 years. But you're going to watch my bacteria get rid of that in just a few hours, 12 hours, and there it goes. And they're respiring a metal. They're breathing that metal, if you like, and causing it to disappear. Now, our Earth's core, iron, for example, is one of the most abundant elements in our Earth's core. So as our Earth was evolving 4 billion, 3 billion years ago, then metals such as iron oxides were a major substrate for respiration. And then various species of nitrogen became substrates, oxidized forms of nitrogen like nitrate, species of sulfur, and then gradually hydrocarbons, organic carbons, also became respiratory substrates. And so bacteria evolved that could respire in a range of different ways, whereas we can only respire oxygen. And so there's a huge diversity of respiratory systems for microbiologists like myself and all those who've passed through my laboratory to study. And these different respiratory systems are important in different ways. For example, the respiratory process in microbes are important for many of the biogeochemical cycles on this planet. They drive the nitrogen cycle, very important in agriculture. They drive the carbon cycle, also, of course, very important in agriculture and for production of food and for ourselves. They drive the iron cycle. We've just heard that iron was an early respiratory substrate. The sulfur cycle, it interacts with many other cycles. And all these cycles and the way they interact are important for the balance of our planet, the balance of the geochemical cycles of our planet, and for a healthy planet. And if they come out of balance, then we have issues. And often, man makes these issues through perturbing these cycles through man-driven activities, such as agricultural activities. And one thing we do across the Norwich Research Park is actually study these cycles, all of these cycles actually, and the way they interact in an alliance called the Earth-Life Systems Alliance, which is directed by Professor Colin Murrell. And it's an example of respiration actually being involved in many people's research right across the Norwich Research Park, not just my own research. Another example of where respiration could be harnessed in biotechnology is actually in microbial fuel cells, and we'll look at this a little bit more later on in the lecture. But I said that respiration was like an electrical current, electrons moving from organic carbon into the electron acceptor, oxygen in the case of our cells. But if you can harness some of that electrical current, you can make it do work for yourself. And this is an example of a microbial fuel cell. Emma and Colin did this video, which is just powering a calculator, generating electricity to power a calculator. And that's coming from a respiratory process, the respiration taking place in that microbial fuel cell. And of course, this introduces the possibility of bioenergy, bioelectricity. Now, it's not so impressive, I acknowledge, just powering up a calculator, but you can start to scale these things up. And this is effectively becoming what you might call a bioelectric power station, taking one fuel cell, adding lots of fuel cells together, putting them in series, amplifying the voltage, and creating a little power station where you can replace modules when they exhaust. And this little power station is actually at the Institute for the Urban Environment in China. My colleague Feng here standing next to it to give it some scale. And we're working with them at the moment, looking at how we can take our biochemistry and help develop these microbial fuel cell technologies further. There's a lot of interest in energy materials and developing bioenergy on this Norwich Research Park. We have Wolfson-supported, Royal Society-supported energy laboratories, a bioenergy laboratory here with a fermenter, an energy materials laboratory in chemistry, and we did get a visit from a chap that you probably all recognise, the Prime Minister himself, David Cameron, and our local MP, Chloe Smith, who was showing an interest in the way that we were looking to develop alternative energy sources, renewable energy sources. And of course, as we try to move away from fossilised fuels, interfacing renewable energy sources with fossilised energy sources as we progress forward will be very important, and I think that UEA has an important role to play with that. And actually energy technologies and energy innovation is an important area for the region more generally, with of course offshore wind being a major investment area at the moment. Also, respiration is important in medicine, and here we see an example of respiration being important in a pathogen, Salmonella, which causes, for example, a typhoid fever, Salmonella typhi. And Salmonella actually invades our bodies, often through the oral tract. It's a foodborne pathogen, and goes into our guts. And once in our guts, it then crosses the gut epithelial and starts entering our bloodstream and then invades cells, which we see this Salmonella here doing. Now, our gut doesn't have oxygen in it. It's actually an anaerobic environment. So whereas we need oxygen for our respiration, bacteria living in this gut don't have oxygen. So they are actually living in this gut and doing respiration without oxygen. And there's a lot of nitrate in the gut, and that can be a substrate for respiration. And so any pathogen invading us through this system and going through this gut, which has no oxygen, maybe actually does need to have a way of respiring in the absence of oxygen. And so this respiration process in the absence of oxygen is important to these pathogens. And so if we can understand that, we might be able to combat these pathogens. Now, remember, our gut has a lot of bacteria in it. And in fact, 95% of our DNA is not our DNA, but it belongs to microorganisms living in this gut. So it's actually a hugely important area to understand the microbiology of the gut and the way that bacteria can live in this gut in the absence of oxygen, and the way that respiration can help to support that. Respiration is also important in another area of biotechnology, the development of biosensors, an area that many of you might be quite familiar with. Those proteins that I talked about, those respiratory proteins that are nano-sized, there's one here called the glucose dehydrogenase, are often harnessed in sensors. And a famous example of that is the glucose sensor, the glucose biosensor. Now, many people in the world are diabetic, and they need to measure their glucose in order to help them administer insulin at the correct time. And the sensor, one of the sensors on the market that is used to sense that glucose, is actually a biosensor in which a respiratory enzyme, this glucose dehydrogenase, an electron transport enzyme, is interfaced into the sensor and helps bind the glucose and send a signal to the sensor that's proportional to the amount of glucose in your blood. And this is a multi-billion dollar area. Now, my group used to work on the glucose dehydrogenase and worked with, we had funding from Roche Diagnostics and Diabetes UK. And we were looking at this enzyme and trying to understand how it worked, really, and its specificity to glucose. Because, in fact, the enzymes that were perhaps being looked at for use in the market at the time did recognize glucose, but they also recognized related carbon substrates like maltose. So we sought to understand how they recognized glucose and how it could improve the specificity for glucose. In fact, we were quite successful in doing that and identifying new forms of this enzyme amongst new species of bacteria. In addition, we've also explored other development of biosensors. For example, developing biosensors for carbon monoxide, where we can see the carbon monoxide increasing and the signal increasing, again, using respiratory enzymes. Now, in my time working with biosensors, and I worked with a colleague, David Russell, who actually won the Royal Society for Chemistry Analytical silver medal for his work on biosensors, we did not make a single penny out of the biosensors that we developed. And there's a lesson for people in the audience here, because I said this is worth multi-billion pounds around the world. And in my early days as a scientist, I think I didn't really understand the need to protect the science. So whilst we were working on these rather interesting sensors with big companies, we didn't necessarily think about protecting. These days we're more savvy, and I think it's therefore quite reassuring to you all, I hope, and to my colleagues around the university, that we're now in an enterprise centre and we're in more enterprising days, and of course we've got an excellent research and enterprise office that wouldn't let me make some of the mistakes I perhaps made as a young researcher. So we are now also developing new biosensors based on respiratory enzymes, particularly one for nitrate. And you'll see this time I'm showing no data. This will reassure my colleagues in the Research and Enterprise Office, telling you nothing about it, because I now know that actually you're not supposed to do that until you've protected your research. So hopefully watch this space and we'll see whether we can develop a nice sensor for nitrate. And we've got Tony there, who was doing some work on this, who's probably in the audience somewhere, who's now relieved that I haven't shown any of his data and might yet become a rich man as a consequence. Okay, so therefore, you now know I'm not going to spend any more time telling you about biosensors. What I am going to tell you a bit about is greenhouse gases. And in particular, a greenhouse gas called nitrous oxide. And you've just seen a culture of bacteria there actually inflating a balloon. And what these bacteria are doing is they're taking nitrate from their environment, in this case just this jar, and they're converting that nitrate, which is a soluble form of nitrogen, which we add to the land in agriculture and fertilization, etc., and they're converting that nitrate to nitrogen gas. And that nitrogen gas was inflating that balloon. Now, nitrogen gas is 78% of our atmosphere, and it's inert. It's not harmful. It's okay to produce nitrogen gas. However, these bacteria aren't just producing nitrogen gas. They're producing another gas as well, called nitrous oxide. Its formula is N2O. And nitrous oxide is a potent greenhouse gas. And what these bacteria are doing is they're using nitrate as a respiratory substrate, and they are converting it to this greenhouse gas and this inert dinitrogen gas. And this greenhouse gas is a problem because as we've added more nitrate to the environment, more of this greenhouse gas nitrous oxide is being emitted and is adding to global warming. So if we can understand how this greenhouse gas is produced and indeed how it's destroyed, it will help us understand this issue and this part of the global warming science and maybe mitigate it, maybe do something to combat it. And so that's what I'm going to spend some of my talk telling you about, this particular greenhouse gas. And I'm going to tell you about the work that we're doing on that at the moment. But before we do that, I am going to talk a little bit about history and introduce you to the theologians and the radicals, and explore just a little bit about the history of nitrous oxide in doing that, because it has a rather amusing history. I find it amusing, at least I hope that you will too. So, the theologian. You've met the greenhouse gas now, nitrous oxide. The theologian. The theologian is Joseph Priestley. And Joseph Priestley, 1733 to 1804, was a theologian, but he was also a chemist. And he discovered oxygen, the respiratory substrate that we use. So very much a father of respiration, even though he probably didn't see himself that way at the time. He also discovered nitrous oxide, the greenhouse gas. He didn't know it was greenhouse gas then, nobody did, but also a respiratory substrate. Now, Joseph Priestley was a great scientist, but he was also a radical, or to the minds of some people, he was a radical. He was a theologian. He believed in actually in religious tolerance. He believed in being tolerant of religious dissidents. He believed that they needed to find pathways by which religion and science could coexist and evolve together. And some of these beliefs and these theories were counter to other people's views at the time. And he was vilified and ridiculed. He actually was effectively a son of East Anglia. He had a parish in East Anglia. He then took up a parish in Birmingham, but was driven out of that parish because it was burnt down, because people were so against his views. He did have views that people thought were quite radical. He did support the French Revolution, and he supported the French Revolution around the time that another famous son of East Anglia supported the French Revolution, and that was Thomas Paine. And this caricature here just shows, you can see revolution, perhaps written, if you can't read at the back, it says revolution, they're sitting on gunpowder, there's a devil standing there. And basically, there were people who took issue with their views. Thomas Paine, of course, went off to America and actually was many considered the father of the American Revolution. Joseph Priestley was driven out to America. He was hounded out of this country and eventually took up residence in Pennsylvania. But I think it's quite interesting that Joseph Priestley is someone that I consider a father of respiration. And in this university, of course, we've got the Thomas Paine Study Centre. We've celebrated a kind of child of East Anglia. And I think Joseph Priestley has a very important place to play in science and actually in humanities and politics of the time. And therefore, I think that if I ever manage to actually build a science and humanities building on this campus, and I hope we'll be able to, I will make a strong case for calling it the Priestley Study Centre. Now, when Joseph Priestley discovered nitrous oxide, like I said, he didn't know it was a greenhouse gas, but people soon found something to do with it. And in fact, the earliest use of nitrous oxide was as a social drug. Of course, we all know it as laughing gas. And it was used in fetes and parties and exhibitions, and this is the grand exhibition of laughing gas. Again, you might not be able to read it, so let me read it. Nitrous oxide, exhilarating or laughing gas, will be given at this particular evening. Gallons of gas will be prepared and administered to all in the audience who desire to inhale it. Men will be invited from the audience to protect those under the influence of the gas. It says laugh, sing, dance, speak or fight. Gas will only be administered to gentlemen of the first respectability. The object to make the entertainment in every respect a genteel affair. I'm not quite sure how fighting sits with the genteel affair, and I'm not quite sure how this image here shows the gas being administered to gentlemen of the highest respectability. They look to be getting that gas under duress there. But this was a big social drug at the time, and people enjoyed it. And actually some of the most surprising people enjoyed it, because this man enjoyed it, Robert Southey. He's the poet laureate in 1813, and he wrote a poem about it, or at least a one-line poem. I'm sure the air in heaven must be this wonder-working gas of delight. I mean, he's the poet laureate. That must be reasonably good. I don't know. I'm creative writing colleagues. Maybe you're here and you can tell me. Some of you might know about him. This man wasn't a poet. This man was a scientist. This is Sir Humphrey Davy, the Royal Society president, 1820 to 1827. He was a scientist, but clearly he fancied himself a poet. And he actually wrote an ode to nitrous oxide. Yet are my eyes with sparkling lustre filled, yet is my mouth replete with murmuring sound, yet are my limbs with inward transport filled, and clad with newborn mightiness around. It's not a good poem, is it? It really isn't. But fortunately, Sir Humphrey Davy was a much better scientist than he was poet. And this little rather famous picture shows Sir Humphrey. There he is. And he's actually producing nitrous oxide, collecting it in these bellows, and administering it to his colleagues in the Royal Institution. Now, this is effectively an experiment on humans. And we're not allowed to do those anymore. Well, not readily. I can't imagine what we'd have to do to get this through health and safety. But there we are. There we are. And you see all these colleagues having a jolly good time experimenting with nitrous oxide. I'm not quite sure why the air's coming out the end there, but who knows. At any rate, one assumes that at some of these events, whether these fetes, these parties, or whether in the Royal Institution itself, that at some point people realized that when they were fighting under the influence of nitrous oxide, they didn't feel the pain. And of course, it emerged eventually that nitrous oxide was an anesthetic. And many people call Sir Humphrey Davy the father of anesthetics because of his role in developing that knowledge around anesthetics. And so, of course, many of us might have encountered nitrous oxide in this sort of scene, though I don't believe I remember there being a teddy there and everyone looking quite so happy when I was administered it back in my childhood. But that's when most of us come across it at its time as an anesthetic. It is a toxin. It is funny, it's a laugh, we can all have a bit of a laugh about it, but it is a toxin. And it's a toxin because it actually damages our vitamin B12 pool. Now, vitamin B12 is important to our metabolism. We can't live without it. In fact, people who are deficient in it will take supplements, vitamin B12 supplements, a big market. And in fact, some people who are exposed to nitrous oxide a lot, like anaesthetists, for example, using nitrous oxide, may have to take vitamin B12 supplements to counter the damaging effect that the nitrous oxide is having on their vitamin B12 pool in their bodies. So it is a toxin. It does have serious deleterious effects on us. And so that's why when these people in 1845 were playing and enjoying nitrous oxide, okay, they didn't know it was a toxin. But these days, we do. And it is coming back as a social drug. So please, please, don't do what this chap did and be tempted. Because it's not good for you. And shortly after this incident, England were knocked out prematurely out of the European Championship. So there you are. There's evidence that it may even affect your performance too. So don't be tempted. Don't go there. If you're at parties and nitrous oxide is being offered to you, say no. Just say no. Now, of course, nitrous oxide is this very potent greenhouse gas, and that's what led us to start to study it. It's 300 times more potent than carbon dioxide, molecule for molecule. It lives in the atmosphere for a lot longer, 500 years here, and it's got a very long half-life. So it only accounts for about 10% of the total global warming impact, if you like. But if we can combat it, then that would be a significant contribution to mitigating greenhouse gas emissions. And nitrous oxide was stable in the atmosphere for many thousands of years. This is 10,000 years ago to about 100 years ago, completely stable. And then, around about 100 or so years ago, we saw a 25% increase in the levels. And that's the time at which we started to we learned how to grab nitrogen from the atmosphere and convert it into fertilizers, the Haber-Bosch process. And of course, it's also the time at which the population started to expand on the planet. And we needed to actually farm more intensively to sustain food for that growing population. So the last 100 years has seen this huge increase in nitrous oxide emissions as a consequence of expansion and intensification of agriculture, and the addition of nitrogen-based fertilizers into the agricultural lands, stimulating the microorganisms living in the soils and then emitting nitrous oxide. So, if we want to then understand how this nitrous oxide is produced and how we might destroy it, prevent it being released, we need to understand the nitrogen cycle taking place on this planet all the time, the biogeochemical nitrogen cycle. And this is what I've been studying for 25 years with many, many fine people around the world and in UEA. And the nitrogen cycle involves nitrate being converted to nitrite in this respiration process, NO2. Then that NO2 minus being converted to NO, nitric oxide. And then that nitric oxide being converted to our greenhouse gas, N2O, nitrous oxide. And then that N2O going to dinitrogen, the nitrogen gas that is 78% of our atmosphere. And that dinitrogen can actually be fixed by microbes again, and other microbes can convert it back ultimately to nitrate. It's a cycle. And we can perturb that cycle through man-made activities, as we have apparently done through adding fertilizer to the lands, arguably one of the biggest agriculture and engineering projects on the planet over the last 100 years. We've done it inadvertently, but we've done it. Now, this nitric oxide here is actually a radical. Not a radical like Joseph Priestley, but a cellular toxin. And it damages cells. It damages the inside of cells. So if a microbe, if a bacterium is producing it, it's quite important that it gets rid of it. And so when it produces it in this nitrogen cycle, it rapidly converts it to this nitrous oxide, this greenhouse gas. Now, the microbe itself, on the whole, isn't overly worried about the fact it's emitting a greenhouse gas or producing a greenhouse gas. It's not sentient, or I don't think it's sentient, and it doesn't worry about global warming. And many microbes have actually done the job they needed to do. They've got rid of the radical, the toxin, and detoxified it to produce a greenhouse gas, which might then simply get emitted into the atmosphere. What's ideal from our perspective is if that nitrous oxide is destroyed, if it's broken down to the inert dinitrogen gas. And there is an enzyme that does this in bacteria, and it's called the nitrous oxide reductase. And this enzyme, small enzyme, nanometer-sized enzyme, it's found inside the bacteria, is really the only enzyme on this planet that can efficiently break down nitrous oxide and convert it to dinitrogen. If it wasn't for this enzyme, our atmosphere on the planet would be nitrous oxide, largely, or in large part, too much, and we'd be a greenhouse. So this planet would not be habitable. And therefore, in my mind, the nitrous oxide reductase is arguably the most important enzyme on the planet. Without it, we'd all be dead, and our planet wouldn't be able to support life, at least not life not human life. And so, just occasionally, in fact, quite often, I'm glad to say, I actually also managed to convince funding organisations that the nitrous oxide reductase is the most important enzyme on the planet. And thank you, BBSRC, because they've supported a lot of this work, and a lot of work in Norwich on this enzyme. And work in Norwich and work with colleagues around the world has led to us having a good understanding of what this enzyme looks like on the nanometer scale. We know the molecular structure of this enzyme. We know its architecture. And through work that a number of us have done, including with Andrew Thompson on the second row there, we understand quite well about how this enzyme works. And actually, the key to this enzyme is copper in the heart of the enzyme. And it's these copper atoms in the heart of the enzyme where the nitrous oxide combines with the electrons and is converted to the inert dinitrogen gas. And so, through understanding this enzyme, we can learn a lot about how nitrous oxide is actually destroyed. And that recognition that copper was at the heart of this enzyme, once we started to know its molecular structure, led us to start thinking, well, now we must understand the impact of copper on the physiology of the microorganisms themselves and understand how they react to copper in their environment. So we went from biochemical studies to microbiological studies. And this is the work that we're doing at the moment and is ongoing in the laboratory, using a molecular detail of the enzyme to inform experiments. And what we have done is to actually culture the microbes in things we call bioreactors, or chemostats we call them, under different growth conditions where we're creating different environments for them. And those environments can vary with respect to how much copper is in the environment, or the pH of the environment, or how much nitrate is in the environment, or whether there's oxygen or not. And we can start to understand the impact of different environmental variables on nitrous oxide emission. And I'm only going to show one experiment of many. I'm not showing many experiments today. This one actually carried out by Heather Felgate, who may or may not be in the audience. I can't quite see who's there. Is that pink hair? So I'll put pink in to sort of go with her hair. So what we have here is an experiment where we are culturing microbes in the presence of high copper concentrations and low copper concentrations, tenfold difference. Now we culture them and we find that actually, regardless of the copper concentration, we can achieve cultures that have the same bacterial populations. There's two lines here. You might not be able to see these two lines because they're literally on top of each other. But the copper replete and copper deplete are supporting the same volume, the same number of bacterial growth. So the bacteria in either condition are alive, they're healthy, they're multiplying, they're able to metabolize. But if we look at nitrous oxide emission now, we see that in the copper replete, plenty of copper in the medium, we can't detect nitrous oxide emission. So the nitrate in that medium, that's the respiratory electron acceptor, is being converted all the way to inert dinitrogen. But in the copper deplete, we can see this line showing increasing amounts of nitrous oxide being emitted as time progresses. So in other words, we have a live culture of bacteria that's emitting nitrous oxide. And that therefore suggests that this organism is responding to a low level of copper by switching off one of its copper-dependent metabolic systems. Now we can do something called studying gene expression, studying the activity of genes. And again, I'm aware of the broad subject coverage of the audience, and so I'll try to keep this straightforward. But our bacteria that we study have got 5,000 genes. And we can look at the activity or expression of each of those genes. And this color chart just shows cold meaning low expression, red meaning hot expression, if you like, and the colors in between are a sort of scale. And of the 5,000 genes, when we compare the activity of these genes in copper deplete versus copper replete conditions, only 50 or so change significantly. So that's a tiny change in response to this different copper levels. And significantly, one gene activity goes down from copper replete to copper deplete. It goes from red, from hot to cold. And that gene is the gene that codes for this nitrous oxide reductase, the enzyme that destroys nitrous oxide. And so what this is telling us is that as we take copper, deplete copper in the system, this enzyme, nitrous oxide reductase, the most important enzyme on the planet because it destroys the greenhouse gas nitrous oxide, actually is downregulated. Its activity is downregulated and it gets taken out of the cellular system. And as a consequence, nitrous oxide becomes the product of this nitrogen cycle. And now this bacteria is emitting a greenhouse gas. So therefore, what we need to do is understand better how this activity of this enzyme and this protein is regulated in the cell, and indeed how the activity of the enzymes that actually lead to the synthesis of the nitrous oxide itself, as well as the destruction of the nitrous oxide, are regulated. And to that end, we've got a large team of people working at UEA, PhD students and postdocs, looking at the regulation of this cycle and also the actual biochemistry of copper metabolism and copper insertion into these nano-sized enzymes. Now, we do think this is going to be important in the environment because 40% of European agricultural soils are copper depleted. And in fact, many agricultural soils around the planet are copper depleted. And that's because over the years, we've replenished our soils, say, with nitrogen and with phosphorus, but we've ignored the minerals, we've ignored the metals, and we ignore those at our peril. Now, this poor lamb is suffering from something called swayback, sheep swayback. It can't stand, it's wobbling. The reason it's got swayback is because it's copper deficient. The reason it's copper deficient is because the milk that it's getting from its mother is copper deficient. And the reason the milk in its mother is copper deficient is because it's grazing, its mother is grazing on copper deficient pastures. So copper deficiency is real, and it's real around the world. And it will only get worse unless we start to actually deal with it. And of course, you might imagine then, if in a laboratory we've established that copper deficiency leads to nitrous oxide emissions, then we need to start taking that observation from a laboratory, from a model system, into a field system. And you'd like to think somebody would fund us to do that. And the good news is that they have, and I'll explain who that is in a minute. But we found that the problem is even worse than we thought, or the potential problem is even worse than we thought. Because I said that 50 gene activities changed, and I come back to the other ones that I didn't mention before. We saw that a number of genes increased in their activity, increased in their expression. And when we analysed what they were, we found that they were indicating to us that the bacteria, in response to producing nitrous oxide, were becoming vitamin B12 deficient. And they were switching on genes that would enable them to live without vitamin B12. And of course, I said to you earlier on that we know that vitamin B12 is destroyed by nitrous oxide. So our microbes can actually synthesize a whole new metabolic system which enables it to live in the absence of vitamin B12, and that's what it's doing. It makes sense because this microbe is a microbe that makes nitrous oxide, so it makes sense that it protects itself against it. However, we can't live without vitamin B12. We're not as clever as the microbes. We need vitamin B12. Microbes, this bacterium, can live without it. It can switch to another form of metabolism. And many microorganisms and soils can't live without vitamin B12. And so if you've got microbes and soils that are emitting nitrous oxide, but are clever enough to have what we might call a vitamin B12 independent metabolism, because it knows the nitrous oxide, I said they're not sentient, so I perhaps shouldn't use that word, but because the nitrous oxide kills vitamin B12 dependent metabolism, it can switch on a new form that enables it to live without vitamin B12. But if an organism is in the environment that can't live without vitamin B12, that is entirely dependent on vitamin B12, it will be killed by the nitrous oxide that another organism is producing. And we can show that happening here because this is an organism sensitive to nitrous oxide, being killed by nitrous oxide, being produced, being added to the system, but then rescued when you inject vitamin B12 into the system to actually replace the vitamin B12 that the nitrous oxide is destroying. So nitrous oxide genuinely can kill other organisms because it hits its vitamin B12 metabolism. So we know that this nitrous oxide emission is not just a greenhouse gas, but it's a biocide. Bad for the environment, potentially damaging the life community around those microbes that are emitting it. So we get a double whammy. There may be solutions. One solution could be that we need to think about new agricultural practices, new fertilization regimes where we supplement fertilizers with copper. Fertilizers perhaps are thought by some to be a bad thing. I happen to think that we need to maximize the productivity of agricultural lands to help support the growing populations. If we don't do that, we'll end up having to chop down more rainforests and then making new agricultural land, and that would be terrible for the environment. So instead, we perhaps have to accept that we need to make the land we've given over to agriculture more productive and make sure that it's not emitting greenhouse gases. So we look to new fertilization regimes which might involve application of copper supplements. And I said that we would achieve funding to take things out into the field, and we have, from the European Union, now set up a nitrous oxide research alliance, Lars and Asa leading that alliance, a number of collaborators all around Europe working together to look at different aspects of the nitrogen cycle, the biochemistry that we do, the microbiology, but also fertilizer companies who will actually work with the scientists and actually see whether we can take some of these observations into field systems and actually translate them into solutions, translating the science into application and hopefully begin to come up with strategies for mitigating the release of this greenhouse gas. So that's the greenhouse gas. I want to talk a little bit now, very much more briefly, about the radicals again, and that chemical radical nitric oxide. Now, I mentioned that Salmonella was invading, let's just see if we can get this going again. I mentioned that Salmonella, when it invaded our cells, was going to be dependent on respiration. And one area that we're interested in is actually studying the importance of nitrogen cycle respiratory systems in Salmonella for pathogenicity. Now, when we're invaded by a pathogen, we have a defense system. And that defense system includes white blood cells, or macrophages, which ingest the pathogen when it gets into our bloodstream and kill it, or try to kill it. And the way they try to kill it is to actually attack it with nitric oxide NO, the radical that I mentioned earlier on. Now, if a pathogen is going to survive the attack by the nitric oxide, you might think it has a defense mechanism to survive. And we know that pathogens do survive because we are susceptible to them. We get food poisoning. People suffer from Salmonella typhi infections. And what it turns out is that Salmonella actually have these respiratory reactions that I've been talking about. They have enzymes inside them that can catalyze the reduction, the conversion of nitrate to nitrite, and nitrite to the nitric oxide radical, and the nitric oxide radical to the greenhouse gas, and now we know biocide, nitrous oxide. And so whilst the macrophage, our blood cell defense system, our white blood cells, attack the invading pathogen with nitric oxide, the pathogen is prepared to deal with it because it can convert that nitric oxide it's been attacked with to this nitrous oxide, which actually in turn could damage the host's vitamin B12 pool. So, as ever, the bacteria can outwit our attack system. So we wanted to study this more and started to take a look at Salmonella. And we wanted to actually establish whether these reactions were indeed taking place. And we found that we could measure large amounts of nitrous oxide production from various pathogenic Salmonella. So they do take place, and they take place quite extensively when we have nitrate in the Salmonella's environment, and there's nitrate in our gut. So this is important. So then we thought, well, can we establish whether this nitrous oxide is actually becoming an aggressive act against the pathogen that the microbe is invading? So we wanted to actually perhaps then see, for example, is this vitamin B12 being attacked within the macrophage when the Salmonella is invading? Well, we don't have a direct answer to that yet, but what we actually did was infect macrophage cells with Salmonella, and we found that when we knocked out the vitamin B12 independent metabolism, so they were completely dependent on vitamin B12, they couldn't colonize a macrophage as well as the parents. This is 24 hours colonization of the macrophage, and you can see that when you have a vitamin B12 dependent enzyme, bacterium, it doesn't colonize very well. And that suggests that the nitrous oxide that's being produced is actually killing the vitamin B12 pool and preventing infection. So an early suggestion that there is a link between infection and vitamin B12 metabolism. Also, we have knocked out some of the reactions that lead to the production or consumption of nitrous oxide. And we have mutants that also don't infect the macrophage very well. Again, suggesting these are important reactions. So once we've established they're important reactions, of course, we have possibilities of targeting these reactions in therapeutic approaches to help prevent or help cure invasion by these pathogens. And so that's a new line of research that we can begin to pursue. And it's quite timely because recently you will have seen announcements that we've been awarded funding with colleagues across the Norwich Research Park, including the Institute for Food Research and the Norfolk Norwich University Hospital, to build a new centre for food and health. And one of the focuses of that centre will be studies of gut metabolism and how food and diet impacts on that. And of course, in the gut, we know 95% of the DNA in there is bacterial. So the bacterial, pathogenic bacteria and friendly bacteria will be an important part of the studies in this centre. We're all very excited about that, and it should be open by 2018. Finally then, just to close off, last 5 minutes or so, the bioenergy and the Martians eating metals. I showed you this video earlier on, and this is the one that shows the microbes. Oops, let's see if we can get it back. Sorry. Technical problem here. Here we go. And this is showing the microbes respiring this metal. And these microbes, like I said, were active probably 3 billion years ago on this planet. But we're studying them now, and we want to actually begin to understand how they're actually doing this process of using metals as respiratory electron acceptors. And we want to understand it because we've seen that these bacteria can be harnessed in microbial fuel cells. I won't play the video again, you saw it earlier on. But if you think about an electrode in a fuel cell, an electrode is a solid substrate. A rock, a mineral of iron, is a solid substrate. So if a bacterium is capable of respiring a rock, a mineral, if you replace that mineral with an electrode, the bacterium is now respiring the electrode, using the electrode as a respiratory electron acceptor. So if you can understand the way the bacteria interacts with a mineral, you will be able to better understand how microbes interact with electrodes in these fuel cells and maybe be able to make them more efficient, work better, work for longer. Now I come back to Mars because there are questions about whether there's life on Mars. And of course, Mars is known as the red planet. And it's the red planet because actually it's covered in iron oxide. That is iron oxide. And there is a school of thought, and some people believe it, some people don't, that actually if there is life on Mars, or if there ever has been life on Mars, it might have been life on Mars that involved using iron oxide as in respiration, as a respiratory substrate. And some of you might remember there was once lots of excitement about images of Martian meteorites with things that might look like fossilized microbes that it was argued was evidence for life on Mars. And if there was life on Mars, it's likely that that life would have been driven by redox reactions of the iron cycle, oxidation and reduction reactions. And one reason why people argued that there was that these microbes are surrounded by a mineral called magnetite. And that magnetite had the form of magnetite that you find around microbes on this planet that are respiring iron. It was the form that is associated with biology rather than simple geochemistry, very ordered magnetite. And there's a view that when you look for life on other planets, you should look for things that shouldn't be there, because you're not going to see the life itself. You should look for things that shouldn't be there. So I was once actually part of a team that was planning the experiments that were going to be on the next European space mission to Mars. I was advising on the microbiology. That was the Aurora space mission. And so you need to look for things that give evidence that life has disrupted the norm. And this magnetite around these shouldn't have been there. If you think of our planet, it shouldn't have as much oxygen as it has. So someone looking at our planet from the outside, taking some sort of infrared study of it and seeing lots of oxygen would say, that's odd. But the oxygen's there because life produced it. So it's a sort of evidence that something's been disrupted. And that's what you need to look for. And that's what people are sort of looking for when they look for life on Mars. The jury is still out, but in principle, it could be there. Coming back to this planet, this iron cycle is important in driving the carbon cycle on this planet because it's the fourth most abundant element on the planet. And that carbon cycle is important for all these other cycles. So it is critical to our biogeochemical cycling. We got money from the US Department of Energy to study this. Again, many laboratories around the USA, but focused in Seattle with these guys leading it from the Pacific North National Laboratory. And one reason they were interested in it was that there's a problem, an issue here on the Columbia River in Washington State with radioactivity, buried radioactivity, mobilizing in the subsurfaces. And the microbes that we were working on that could use iron as respiratory substrates can actually influence the mobility of radionuclides through these subsurfaces and perhaps help stop the radioactivity going into nearby rivers. So we were interested from that perspective, but also began to get interested in it from the point of view of the microbial fuel cells. And the question that we wanted to ask was, how do electrons leave a cell and find their way to a mineral, to a rock? When we were looking at the nitrate respiration that we talked about earlier, nitrate is soluble and can come into a cell. But how do electrons leave a cell to find a rock or an electrode? Well, we identified with our colleagues around the world some proteins on the cell's surface that were in the right place to make electrical contact with an electrode or with a mineral. And when they were knocked out using genetic techniques, the capacity for iron respiration was lost compared to the parent. So we were looking at the right things. We did biochemistry. we take these proteins and we make crystals from them. And then we use those crystals to get molecular structures using X-ray diffraction. Again, colleagues around UEA, very clever colleagues, experts in this area. And therefore, we can amplify this little protein image to a molecular structure at the nanometer scale. Takes 10 years, by the way. It looks as if we just did it in a few minutes. It takes 10 years. An amazing piece of work. And this is a protein. This is a polypeptide around the outside. And in that polypeptide, you can see molecules, and they're molecules of iron. You can just see them as they go around. And they're bound within things that we call hemes, very similar to the hemoglobin in our blood. And they're red, and that gives the protein this red color. But this protein is a molecular scaffold that's holding these ions a few nanometers apart in the scaffold. Wire is made of iron. And what this actually is an electrical wire insulated by a protein sheath, just as a wire is insulated by a sheath. So in fact, what we had discovered was a nanowire that would conduct electricity through this insulated protein. And this was very exciting. And in fact, we moved from there to develop models, which we're in the process of trying to prove, that show how this nanowire would actually pass through a biological membrane to move electrons from inside of the cell to the outside of the cell. And we're approaching the end now, and I'll just therefore finish off with this video and one more slide afterwards. So this is the video of a nanowire. This is a cell membrane, and we're just stripping the membrane away, and we can see this is a protein sheath. This is on a nanometer scale, 10 to the minus 8 meters. Just take all the membrane away completely, and we can see this protein insulating sheath. Soon that's going to be stripped away. We're just stripping some of it away now, and we can start to see those molecules, those red molecules of iron on the scaffold. We just take all the protein away now, and we can see the wire. These molecules of iron just far enough apart to allow electrons to hop from one to the other, and now back in the membrane, and we see the wire going through the membrane. Of course, it's in reality insulated by a protein. So a nanowire. Electrical conduction on the nanometer scale. And those nanowires would sit across these membranes, explaining how you can conduct electrons out of the cell to a solid substrate. And we can produce structures, molecular structures, where we actually have mimics of solid substrates attaching to the protein, and you can see that they're just getting close to the terminus of these wires that go through the protein, so an electron can hop from the wire onto the mineral, if you like, that's sitting next to the wire. So that's the solution to this problem. And so I leave in the final slide with the frontiers of catalysis, because having identified the molecular structure of nanowires, having purified them, we can now start to try and get new work out of them and go into new frontiers. Because we can now move electrons, we can now make electrical currents going back and forwards. We can control the direction of flow into or out of cells. We can explore ways in which we can link those wires to semiconductors, which can be excited by light to make electrons jump onto or come off the wires. We can potentially directly ground the whole system to electrodes or interface electrodes with light-driven dyes and semiconductors to help harness light energy to move electrons into and out of the cells. And then we can start harnessing the cells and engineering the cells to produce cell factories. Cell factories that could potentially potentially produce hydrogen using protons. That can actually be sinks for CO2, converting CO2 to a fuel called formate, but doing carbon capture as part of that, stripping CO2 out of the system. Making platform chemicals that are the basis for many chemical syntheses. Maybe destroying greenhouse gases like nitrous oxide that I started off by talking about. And we can also use these bacteria to produce the semiconductors that actually could be involved in these systems. This is an image of a bacterium called Klebsiella, which has actually got cadmium sulfide semiconductors on its surface that we've made it synthesize itself. So we can start fusing materials chemistry with biological chemistry to help develop these systems. I had a conversation the other day. Those there will know the conversation that maybe said that materials chemistry and biological chemistry were not compatible. They're perfectly compatible, and this is how you bring the two together. And I think this is an area where we can be very powerful at UEA and on the Norwich Research Park in the future. So that would bring us to the end. And in closing, I would say we met the greenhouse gas nitrous oxide. You've seen the opportunities for bioenergy there at a number of moments in the talk, not least at the end. We met the theologian, we met the radical, we met the laughing gas. Well, we sort of met the Martians, maybe didn't quite, but I leave that up in the air. And we saw the bacteria eating the metals. And I did give you very much a perspective. I haven't given you much of the history of 25 years of science in my group. And so the pictures I was showing on the way are PhD students and postdocs who are currently in the laboratories around UEA working on these, I think, frontier projects. There are many, many people to thank from the last 25 years. These people I've all held research grants with. They've been joint grant holders, and I thank them all. At the moment, I'm kind of mostly working with Julia Butt, Tom Clark, Andy Gates, and Gary Rowley, which we hold, with whom we hold a lot of research money. But I've worked extensively with all these other people. Too many to name, but they know who they are. And thank you to you all if you're in the audience. And apologies if I've forgotten your photograph if you're in the audience, because there's a lot of people. Every one of these people, every one of these research groups would have many, many PhD students and postdocs over the years. And if any of those are watching or online, thank you to you as well. But most of all, thank you to these people. And because none of it's ever possible without your family. And I know that my dad is watching live, because this has been streamed live. So hello, Dad, and thank you. My mum went upstairs about 5 years ago, but I know she's with us in spirit. So thanks, Mum, as well, and thank you all.
So thanks very much, David. My name's Neil Ward. I'm the other Pro Vice-Chancellor, PVC for Academic Affairs, and it's my job to just hold the ring for some questions. David's agreed to take questions from the audience. We have a roving mic, which is over there. So can I invite anyone to ask a question of David on his lecture? Now, we did this lecture 2 weeks ago in London with alumni, and loads of hands shot up. There's one there. And could you just hold on until you get the mic?
Hello. David, you know I'm a computer scientist. And I got all excited by your groovy hybrid organisms at the end that had sort of semiconductors patched on the side of them. What's one of the things you need to make a good computer is speed. Could you talk a little bit about how quickly those electrons could zoom from one to the other, and then just dwell a bit on the switching? Can you, because you also need that to make a biocomputer.
So the electrons are moving at about 1,000 electrons per second per molecule, and that's a very fast rate of electron transfer, and that's quite critical because actually the biological rates of respiration actually have to be fast. So there's certainly potential for engineering those wires and building them up. And I mean, this is something we've talked about exploring, because what we could do is, take a 16 nanometer nanowire and think about doing gene fusion to make one twice the size and one 3 times the size and one 4 times the size, and so on and so forth. So you could actually start thinking about laying wires down on chips and things like that. And then, of course, when it comes to sort of breaking the current, there's all sorts of ways one could do that. You could engineer a sort of light-sensitive hinge into the protein. So within the wire, two of those iron molecules, they're about a nanometer apart, let's just say. And if they became 2 nanometers apart, you would break the current. So if you could put some sort of switch in there, maybe a light sensor switch that just broke the protein. For example, I don't know if I'm going to get too technical, but you can have things called disulfide bonds in proteins, and they would hold two regions of the protein together, and you could break the disulfide bond, it would open up, and you could reform it, it would close. So there's ways in which you can engineer a molecular break into it that would then break the current. There's also ways in which you could think about how you might put the current one way and not the other, but that's driving forces. But yeah, it's all there to be done, so let's do it. Though we've just given it away because now we've just lost the patent.
Any other questions? There's one right at the back, Ollie.
So I've been definitely, very excited about seeing all this issue about seeing how biological fuel cells could actually be out there and being able to be used for producing electricity and all that. And I've been wondering, how far are we and what is the capacity for, let's say, seeing in the next 10, 20 years, will these fuel cells be able to produce enough electricity for something? Or,
Yeah.
I mean, because as far as I understand right now, we're talking about very small currents, isn't it?
Yeah. I mean, all things, these sort of stages of technology start off small, and then you have to see where they go. And if you go back to, mid-1800s and you think of some of the things they were playing around with then, even the early days of electricity compared to where we are now. So you have to hold your nerve on these things, and this is my reason why I always think that you need long-term funding programs. But there's a lot of groups around the world, and we're working with some of them that are trying to sort of see how far you can push the technologies. I showed you an example of a little power station whereby you can put sort of fuel cells in series, and then you can actually build up the voltage that way and increase the currents. They're never going to be as efficient as a gas-fired power station or a nuclear power station, but you will find uses for them. For example, in rural communities where there's a lot of waste materials, you could be powering houses or particular kind of things that need powering in those sort of environments. Subsurface environments where there's lots of organic sediment and lots of electron acceptor, where you might want to put electrical devices and then have them powered actually out of the environment themselves rather than send wires down to them. The seabeds, again, organic rich sediments, lots of seabed electrical devices for which you can't power them with solar because the light's not getting down there, but you could actually harness the organic material and the electron acceptors there and get electrical currents out of them and power the devices there. So there's going to be a lot of niche applications, and potentially in some areas of the world you might find that they're actually a good genuine substitution for other energy sources.
Any more questions?
David, could you expand a little bit on the work that I think you called it the Nitrous Oxide Research Alliance, NORA,
Yes.
is doing? The reason I ask is because you said that you can certainly add copper to fertilizer, but as you will know better than me, it's very easy to increase copper toxicity in soils to the point where you can actually cause grazing animals to keel over as well. And also in veterinary medicine, of course, copper is used as a biocide. So there's a very delicate balance there.
Yeah, there is. And that's exactly why you need to be working with fertilizer companies, but also agriculturists and soil scientists to help you to actually take these things into field trials. So just to expand for other members of the audience, copper is actually used as a biocide itself in some areas. And so, like a lot of metals, you can have a range where the metal or the mineral is bio-beneficial and an area where it's biocidal, and it's a fine balance. And the actual bioavailability of the copper in any particular soil will also be dependent on the structure of the soil, the pH of the soil. And so what works for one soil type might not work for another soil type. And so that's why you need to, to do a lot more experimentations, why you can't simply go from my model system in the laboratory and start throwing copper around the fields. So, of course, that's where you do need the sort of alliance that the Nitrous Oxide Alliance is. That does bring together the biochemists like myself with soil scientists and with scientists and fertilizer companies, so we can access sites. We do have access to sites that do have copper deficiency and can start actually doing some careful experiments to see where it goes. There are some areas already that are being fertilized with copper. It's quite normal in the wine industry in France. So it's not unheard of, but one needs to get these things right. And I mean, I mentioned pH, but pH will be one critical one, which does affect metal speciation in quite a big way.
Any more questions? Okay, well, I think wine is waiting for us outside, so we should probably draw things to a close. I think thanks, David, for such a panoramic talk. It took us to Mars and over a time period that spanned about 4 billion years, I think. And I think we should just thank him for his excellent inaugural lecture in the usual way. Thank you.